Hello! Welcome to our video on an overview of proteins. The learning outcomes for this presentation are to 1. Briefly state what proteins are. 2. Describe the structures and functions of proteins. 3. Briefly state what peptides are. 4. Describe the 4 types of protein structures. 5. Describe the common shapes of proteins. 6. Explain what enzymes are and how they function. 7. Describe what glycoproteins and proteoglycans are. And 8. Describe what cofactors and coenzymes are. Proteins are the most abundant and important organic molecules of the human body. They contain the basic elements that carbohydrates and lipids do, which are carbon, hydrogen, and oxygen, and they also characteristically contain nitrogen as well. Proteins are polymers made up of amino acids, their monomer. There are a total of 20 amino acids that can rearrange to make a wide variety of proteins. There are 7 major functions of proteins in the human body, and many of the functions listed can be applied to all living organisms. Proteins are important for structural support. There are many different proteins embedded in our bones, muscles, and skin that provide us with support, such as collagen. There are also proteins that play a role in movement. Actin and myosin are muscle proteins that are vital in the contraction of skill. Proteins are also used to create mechanisms of transport across the body. An example of transport proteins are the channel proteins. Channel proteins are embedded within the plasma membrane and allow for the flow of ions, fluid, and other molecules. Proteins are also essential for the regulation of metabolic processes. Enzymes are proteins that lower the activation energy needed for metabolic reactions. An example of an enzyme is lactase, which facilitates the catabolism of lactose into glucose and galactose. Proteins are also important in controlling homeostasis and coordinating the body systems. Examples of these types of proteins include protein hormones, such as insulin, which is responsible for the regulation of blood sugar levels. Buffering, such as the regulation of the body's pH, is another important role that proteins play. By binding acids or bases, proteins can neutralize the blood plasma pH to keep it within the normal range. Lastly, proteins are important for the immune response. Antibodies are proteins that aid in the adaptive immune response needed to target specific pathogens and neutralize them. 2 common antibodies in infections are IgG and IgM. The structure of proteins vary from one protein to the next. However, they all have some characteristics in common. All proteins can be described as having long chains of amino acids. Each amino acid consists of the following: a central carbon atom, a hydrogen atom, an amino group, NH2, a carboxyl group, COOH, and a variable side chain, or R group. Peptides are short chains of amino acids connected by peptide bonds. The linking together of amino acids requires a dehydration synthesis reaction between the amino group of one amino acid and the carboxyl group of another amino acid. When a covalent bond forms between the 2 amino acids, it is termed a peptide bond and results in the formation of a peptide molecule. When 2 amino acids bond together, it is called a dipeptide. When 3 amino acids bond together, it is called a tripeptide. Polypeptides are tripeptides and larger peptides. There are 4 types of protein structures: primary, secondary, tertiary, and quaternary protein structures. Primary protein structures refer to the sequence of amino acids along a polypeptide. Secondary protein structure takes the primary protein structure, and because of the different charges, polarity, and molecules, bonds can form, creating spirals or pleats, also known as helices or sheets, respectively. Tertiary protein structures take the secondary structure and combine the helices, sheets, and irregular folds into a 3-dimensional structure. Lastly, the quaternary protein structure is the final protein complex produced by interacting tertiary protein structures. Common shapes of proteins include fibrous and globular proteins. Globular proteins are in the form of soluble spheres whose shape is based on the tertiary protein structure. Fibrous proteins are in the structural sheets or strands whose shape is based on the secondary or quaternary protein structures. Important proteins that function within the human body are enzymes. Enzymes are proteins that catalyze or speed up chemical reactions by lowering the amount of energy required to undergo a reaction. Enzymes can be utilized more than one time and can facilitate multiple reactions prior to deteriorating. Substrates or reactants bind to an active site on an enzyme to create products. Enzymes exhibit common characteristics. 1. Specificity, meaning that the enzyme can catalyze only one type of reaction typically. 2. Saturation limits, meaning that once the enzyme becomes saturated and the active site is occupied, it cannot bind to another substrate until an active site becomes available. And 3. Regulation, which refers to other cellular chemicals or molecules that regulate the activity of the enzyme to increase or decrease its activity. Enzyme function can be affected by many factors. Two common factors are temperature and pH. An extreme change in either direction of temperature or pH can lead to denaturation, which is a change in the shape of the enzyme and loss of function of the enzyme. In this figure, we can see how the enzyme and substrate interact to facilitate a reaction. First, the enzyme binds to the substrate at the active site. Once bound, the enzyme changes its shape slightly, creating an enzyme-substrate complex. The change in shape conformation of the enzyme catalyzes the reaction to change the substrate to the product. Once the products are created, they are released from the active site of the enzyme. This reaction can be catabolic, as seen in this figure, where the substrate is broken down into smaller subunits. Likewise, the reaction can be anabolic, where more than one substrate can bind to the active site and build a larger molecule from the smaller subunits. Proteins can combine with carbohydrates to form molecules that are both proteins and carbohydrates. Examples include glycoproteins and proteoglycans. Glycoproteins are composed of large proteins combined with small carbohydrates. Examples of glycoproteins include enzymes, antibodies, hormones, and components of the plasma membrane. Proteoglycans are composed of large polysaccharides, also known as carbohydrates, combined with polypeptides. Examples of proteoglycans include hyaluronic acid and heparin. Have you ever wondered why taking a multivitamin is important and how it works? Vitamins and minerals are also known in science as coenzymes and cofactors, respectively. Cofactors are ions or molecules that bind to an enzyme before substrates can bind to facilitate the binding of the substrate to the active site. Coenzymes are non-protein, organic cofactors that also facilitate enzymatic reactions. In this presentation, we learned that proteins are macromolecules that have carbon, oxygen, hydrogen, and nitrogen, that they are made up of amino acids and have important functions in the body, such as structural support and regulation of metabolism. We also learned that peptides are small chains of amino acids and that proteins have 4 structural levels of organization: primary, secondary, tertiary, and quaternary. Proteins also have common shapes in the globular or strand form, and their shape affects their functions, as we saw in how enzymes function. Lastly, we learned about molecules that are formed from combining with proteins or that interact with proteins to control and regulate bodily functions. On behalf of the Academic Success Center, thank you for watching our video on proteins. For more informational videos, please visit our YouTube channel at Ask ASC. Thank you, and have a great day.